Hello. Well, I'm Eneas. Thank you very much to the organizers for inviting to be here and for organizing this great conference. And well, the title of my presentation is "Can we transplant the soil microbiome between two systems?" And I want to thank David Alcaraz because he already answered it. So goodbye. Yeah, no, I'm so glad that we are converging in our interests and ideas. And well, this is going to be in the field. I want to talk to you a little bit about milpa agroecosystems. Milpas are, as we understand it, are the traditional family agriculture of Mesoamerica, including most of Mexico and Central America, and there are even milpas in the United States and in other parts of America. But well, and we believe that they are the direct ancestors of the scenario of domestication of several important species like maize, like beans, like squash, probably chili, and many others. But those are the most important ones, especially mice and beans. With the intensification of agriculture, these polycultures are rarer and rarer, as Luis David believes that they are extinct. But I believe they are in the process of going extinct, because they are not so easily commercialized. So their products, they are very productive in terms of calories, but it's very difficult to sell something that varies along the year and that is produced in very different quantities. For example, in Hidalgo, everyone that has water for irrigation doesn't do milpa, they do intensive agriculture, but there are still many families that do it, and there is also a lot of diversity in milpas. We wanted to first to characterize the microbiome of the milpa system in central Mexico in different environmental and cultural conditions. So we chose a gradient that is not too long. It's like 100 kilometers, and but there are huge variations in the climate, the vegetation. For example, these are forests, but here is a little bit of shrublands. But these are forests, but it's really dry. This is a very humid cloud forest, and this is jungle. Okay. Okay. Well, here you can see the precipitation gradient that is very steep, from very low precipitations to really a lot of rain. And temperature also varies, rating in transit. And this area is maybe Otomi people. They call themselves Ñañú. This area is Sierra, where the people are mestizos, the typical Mexicans that speak Spanish and have a mixed ancestry. And this area is Huasteca, and they are Nahua speaking. So there is they also have differences in management. I'm not going to talk about management or cultural differences in the gradient, but well, that's for another talk. We sampled 28 milpas. They don't look so bad as the pictures we saw earlier. But in the upper part, they do look like that. And here in this one, it's not so easy to find a very good, successful milpas, but there are some. And we took 8 samples in the root zone of maize. So it's not rhizosphere per se, but it's in the root zone. And we obtained 1.7 terabytes of compressed sequence files. So many bases, many hundreds of millions of bases, and we sampled lots of metadata. This field campaign was done by Heriberta and myself. And Heriberta is a now speaking producer of milpa, who's also an engineer in biotechnology. So I have collaborated with her a lot, and we collected many metadata, including geolocation, soil analysis, one per milpa, so 28, and one survey per milpa. We also identified the plants. With Heriberta identified the plants, and I only wrote it down as she was dictating me, because she's very good at identifying plant. And we did some experiments with plant physiology here. Here we are measuring electron transport with plant fluorescence, but those data are also for another day, because it's a lot. And Matteo helped me a lot with bioinformatic analysis. Here is the distribution of phyla in the 28 milpas. These are averages per milpa. And as you can see, it's quite stable. I mean, well, these phyla are very big groups, but the dominating phyla are the same. That is something that we have observed in all milpas we have studied. It's also a common distribution of microorganisms in agricultural soils. So there's nothing there, but the devil is in the details. Well, even at the genus level, there is here Spanish. There is quite some stability with the most common species genera being some actinos and gamma, well, Pseudomonadota, etc. But well, this only gives us a very superficial view of the communities. But well, I wanted to focus on the effect of the soil properties on these microbial communities. But we have too many variables, like 40 soil variables. I don't know. So we chose some, and we did factor analysis to get a small set of variables that we could make linear regressions on. And well, this is just to show at the community level. This is data diversity. So here we have Breit-Curtis distances, and this is the distances, the Euclidean distances between the pairs of elevations, right? So we took two elevations, we calculated the distance, and Pythagoras theorem, and we plotted this, and well, I heard somebody call 0.3 a low correlation, but in field ecology, it's 0.16 and significant. It's quite good. And this one is even better. This is the Breit-Curtis distances versus what we call soil structure. I apologize for the names. I don't know how to name these factors. Well, we said soil structure is organic matter, sands, clay, and field capacity. These are the three are orthogonal, so they really measure different things. These soil chemical properties, pH, calcium, and conductivity. This is how alkaline is the soil, but it's not alkalinity, because alkalinity has another definition. But it's high values of these are very alkaline soils. And these are all nutrients. Right? So this is the soil structure had a strong effect. Soil nutrients had a really huge effect, with 0.6 and Pearson correlation. But we wanted to study the effects on the different microbial groups. So we did general linear models with mixed effects models with milpas as random effects and the factors as fixed effects. And we can see that there are even phyla level effects of the soil conditions. And I would say elevation is a proxy for climate, right? Because, well, we don't really have a measure of climate, but it changes a lot with elevation. So elevation integrates several things, but one of them is climate, one important one. So we can see very different behaviors. For example, this is cyanobacteriota. So the cyanobacteria, they have positive correlations with soil nutrients, but negative correlations with elevation, with soil structure. Surprisingly, Acinetobacteriota, I will show you why in this case, didn't have lots of noticeable effects, verruco microbiota neither, etc. And here is the what I was telling you about Acinetobacteriota. That some Acinetobacteria do prefer alkaline soils. They are famous for liking low pH, but it's not always the case. Actually, more have negative correlations with how alkaline. The most have negative correlations with how alkaline is the soil, but some have a positive one, and so that's why we don't see phyla level effects. Well, these are interesting things. We also made sparsity network analysis. Sparsity is very good for this kind of data because it takes into account the compositional nature of microbial relative abundance, that is that there is some one. So it's definitely recommended over Pearson or Spearman correlation, etc. And we found a lot of modularity on correlation networks. So they are huge modules. Modules are very commonly correlated with the factors, with our factors from environmental conditions or physicochemical properties of the soil, and they also vary a lot in their phylogenetic compositions. So this raises a question that I unfortunately haven't researched yet of can we use these models as a basis for management of my microbial community? Can we try to target the models, maybe increasing the abundance of some members and see if the others also respond? I don't know. And I'm open for suggestions. And this is also from the same network. And this is which interactions are, well, apparent interactions, because we actually tested this experimentally. But they are, which ones are positively correlated or negatively correlated. I believe some of these are actual antagonistic interactions. Some of these are actual nutrient trophic-like interactions. And some of these are just bacteria that like the same place, that have the same niche. And these are negative. Well, we can see that in the positive ones, there are lots of interactions between strains from the same groups, for example, from the same phyla. Here we can see that here. But there are also quite a lot of interactions between phyla. So it's a very interesting dynamic here. And in the case of a negative interaction, is actinomycetota against everybody else. So actinomycetota is attacking everybody, apparently, and they are known for that. So, can we transplant the soil microbiome between traditional and park agroecosystems? To have a first approach to this question, we set up an experiment. Well, the first time we tried this, it failed because of two reasons. One, it is very difficult in the field to spread microorganisms homogeneously. I felt very bad after doing that. I said, oh no, this is not going to work. And then Tlaloc agreed with me and it didn't rain at all. So Tlaloc is the god of the rain in Mexico for the Aztecs. Then we tried it a second time. This time it did rain. And what we did was we soaked we collected soil from different milpas, and then we soaked the maize with a bit of sterile water and the collected soil, and we planted it. Here we are planting the experiment, and we used 4 donor milpas and then soil from the same milpa as a control. And here is the producer planting, helping us in our experiment. Here I also planted with Iberta, and you can see by the post who's the pro, right? Clearly, I don't know how to use these things yet, but okay. So one thing at the level of beta diversity, you can see. Well, in the triangles are the original communities from the milpas that we sampled, and the circles are what grew from the seeds that we planted. Also the maize root zone, and the triangles was just soil. So you can see there are changes even in the original milpa. We expected this one purple, but also there are also lots of changes in the other milpas, and as you can see, the communities are quite different and quite separated depending where the soil came. They are not necessarily closer to their original community, but because this is very complex and maize is growing and it's selecting different microbes. But we used ANCOMBC to detect significant differences between these are log ratios, and then it's between the soil of the milpa and the seeds that grew in the experimental using the donor seeds and comparing it to the changes with the original milpa soil. So the blue one is like a kind of control. That are the changes that we see when maize is growing, and the other one is the soil that we added from another milpa, and you see that several of them coincide. This is not so easy to explain, but this is suggesting that actually these microorganisms are coming from the experimental milpa and colonizing the growing maize. Let me try again. This one. This is the experimental soil, the seed soil soaked with experimental soil. This is the seeds coming coming from the experimental soil, compared to the sorry. Okay, Here's Charles Chaplin talking to you. Okay. Yeah. So we are comparing them to their own soil through log ratio and to the soil where the control grew. So they are enriched compared to both. Yeah, they came from the donor milpa. Here we have less, but one of them coincides at least at apparent genes level. And here is the other one. We couldn't do this for the 4 because we lost one sample and then we couldn't do the analysis. This is strange that this one had the opposite sign, but in general it coincides. What I want to convey to you is that we could at least alter the communities. I mean, if it was not very clear in these graphs, at least here you can see it really clearly, right? There are very big differences according to which soil was used to sow the seeds and what grew. So, thank you very much. Well, this project was funded by a Kaga Foundation, that is a Swedish foundation that gave us a grant to do this, and Society funds my position, and the milpa producers. Without them, there's nothing. Thank you.
We have a question for the next. Maybe I'll just.
Yeah, there is one there. Okay.
Thank you for the talk. I have a question. Which is the perspective of use transplant, can we say, of the microbiome of one milpa to other milpa,
Yeah.
to have better conditions for the growth of the maize, or to prevent seeds, or what is the perspective of this work?
All of those things. Luis David showed us in the lab that you can even improve drought tolerant using microbiome transplants. So I think it's a very easy economic method to improve the productivity without spending much. You only have to travel to where the donor milpa is and bring some soil. These microbial communities definitely help maize or plants in general to sustain, to live in under stressing conditions that can be biotic or abiotic. They produce plant hormones that can be very good for the plants, etc. That is, yes, Luis?
I think we will continue with the program, but thank you.